Welcome to today's AAPS webinar. Thank you for joining us. Before we get started, I'd like to take a moment to acquaint you with a few features of this web event technology. At any time, you may adjust your audio using any computer volume settings that you have. On the right-hand side of the screen, you will see the Q&A polling window. There's a large window which holds all of your sent messages, and a smaller text box at the top where you will type in your questions. To send a question, click in the text box and type your text. When finished, click the Send button. Questions will be addressed at the end of the presentation. I would like to turn it over to our moderator, Stacy Royston.
Thank you very much. Today's eChalk Talk is titled, What's Next in Precision Medicine? Small Molecule Biomarkers to Personalize Disease Prediction, Diagnostics, and Therapies, hosted by Sapient. It is my pleasure to welcome today's speaker, Dr. Mohit Jain. Dr. Jain is a physician scientist with over 20 years of expertise in physiology, biomedicine, engineering, computational biology, and mass spectrometry-based metabolomics. Prior to founding Sapient, he formed and was director of the Jain Laboratory at the University of California, San Diego. There, he led a multidisciplinary research team of chemists, engineers, mathematicians, epidemiologists, and physicians to develop next-generation rapid liquid chromatography mass spectrometry systems to probe the non-genetic landscape of disease across population-scale human studies. Dr. Jain founded Sapient in 2021 as a spin-out of Jain Laboratory to expand upon the mission of accelerating human discovery and drug development through the nexus of high-throughput analytical mass spectrometry, computational biology, and population-scale clinical studies. Dr. Jain, the floor is yours.
Thank you so much, Stacy. It really is our pleasure to be here today. And we're excited to take the next half an hour or so and share with you some of our ongoing work in precision medicine, and in particular, how we're using small molecule biomarkers to transform our understanding of human disease, as well as disease prediction, diagnostics, and therapeutics. So to begin, it's really important to frame out what's the underlying problem. And when we think about human biology, for every major discovery, for every question that's answered, this only brings about more questions. And so at least over the course of my lifetime and my continued life, it's likely the case that the unknowns in human biology will far exceed anything that we know. Now, this introduces some unique challenges when we're thinking about how to understand disease, how to diagnose human disease, and ultimately how to treat human disease. Now, when we think about the origins of human disease, this really can be broken down into genetic and non-genetic factors that contribute to disease over the course of one's life. And we can begin to model the percentage or population attributable risk that occurs from your underlying genome, as well as your non-genetic factors. Now, when we do this across tens to hundreds of thousands of twins, as well as many other types of heterodyability studies, this is an analysis that was performed by Steve Rappaport, as well as many others. What you find is that the minority of disease risk, somewhere in the order of about 25 to 30% of disease risk, comes from your underlying genome. What this means is that the majority of disease risk over the course of one's life has nothing to do with your genome. This is true for cancer, heart disease, neurologic disorders, neurodegenerative Alzheimer's disease, lung disease, obesity and metabolic related diseases, as well as autoimmune, maternal health, and other diseases that we think about in human society. It's become clear over the last several decades that while we've learned much about the underlying genetic underpinnings of human disease, it's both internal as well as external exposures that occur over the course of one's lifetime that has profound influence on biological processes, the development of disease or health over time, as well as ultimately treatment responsiveness. Here's a recent analysis of precision medicine by McKinsey and others. And this is a very powerful quote that came from this underlying report, which reads, while genetics is a powerful indicator of disease and reflects our individual variation, it does not necessarily inform on the current biological state, which is the most predictive indicator of health. Current functionality and dysfunctionality is better reflected in the transcriptome, proteome, and metabolome. This metabolome, of course, being the small molecule biomarkers that we spend much of our time thinking about. Now, as I mentioned here at Sapient, we particularly focus in these small molecule biomarkers precisely because they enable a readout of both genetic as well as the non-genetic factors that contribute to human disease. These small molecule biomarkers are synthesized both in normal tissue as well as diseased tissue, as well as within the microenvironment. And given their physical chemical properties, these are actively extruded from the intracellular environment into the central circulation, where they can be measured as a non-invasive metric for tissue or cellular biology. These oftentimes reflect very specific disease processes, whether they be healthy processes or abnormal disease processes. And these are among the most ancient of molecules, highly conserved across species, from cellular systems to preclinical models to in vivo in humans, which allows for great translation of these discoveries, as well as cross-integration across different systems. Now, thinking about these small molecule biomarkers in comparison to more traditional protein and or DNA biomarkers, what we find is that small molecule biomarkers have a much more heterogeneity and variability in their underlying source, meaning they can come from endogenous sources, such as the liver, the heart, the brain, muscle, fat cells, et cetera, as well as the microbiome that lives symbiotically within our gut and our skin. These also come from exogenous sources, our diet, our exposures, everything essentially we eat, drink, or smell, smoke over the course of one's life. This is in contrast to protein or DNA markers that are largely reflective of endogenous processes that occur within cells and tissues. Now, as mentioned in the prior slide, small molecules, given their underlying physical chemical properties, they can be synthesized intracellularly or absorbed from the world around us, and then released into the central circulation where they can be measured. Again, this is in contrast to protein or DNA, which are obviously very important, but read out different aspects of this. Protein absolutely reads out active biological processes, but there's only a very small portion of the underlying cellular proteome that's released from the intracellular space to the blood. These released proteins typically come from the liver as well as immune cells. And again, the vast majority of the proteome as well as protein modifications are retained intracellularly within specific tissues and cells. Similarly with DNA, the vast majority of DNA is housed intracellularly unless there is cellular rupture with extrusion of DNA into the plasma. And as mentioned earlier, DNA is largely reflecting and reading out underlying host heritability. So it tells us a lot about where we come from rather than those active processes that are giving rise to health or disease in real time. When we think about the size of small molecule biomarkers, these are typically quite small. They're below 2,000 Dalton, in contrast to proteins, which are larger, greater than 2,000 Dalton. And given the size of proteins and their three-dimensional conformational states, these tend to be rather unstable, where small molecule biomarkers are among the most stable organic material in nature, given their size and the way they're conformed. Another way we can begin to think about small molecule biomarkers in relation to DNA and protein is as dynamic indicators of health and disease. As mentioned, your DNA is largely set from the moment of conception and really represents a static measure of underlying disease heritability. In contrast, proteins can represent acquired factors, though as mentioned, these are largely maintained within the intracellular compartment and really read out only host factors as opposed to external factors that give rise to disease. In contrast, small molecule biomarkers really represent a functional readout of underlying phenotype. These are highly dynamic measures that can change in response to a number of factors, whether they be host genetics, lifestyle, microbiome and pathogen, environment, medication, as well as many, many other factors which accumulate over the course of one's life. Now, next-gen sequencing has obviously transformed our understanding of DNA markers, enabling massive measure of underlying genetic sequence across large populations of individuals, and in doing so has really allowed us to understand the genetic underpinnings of human disease. The problem has always been that traditional bioanalytical methods really capture a very small portion of the underlying small molecule biomarkers or metabolites that are present in humans. This is where Sapient's transformative technologies and our next generation mass spectrometry systems have really enabled great extension of our understanding of this space, enabling for the first time measurement of the thousands of molecules that are comprised within humans and human biological systems, and doing this across thousands of individuals at a given time, greatly extending the amount of information that we can learn about an individual, as well as the information we can learn about human disease across populations. These approaches are enabled by our what we call rapid liquid chromatography, or RLC, mass spectrometry technologies that enable us to map small molecule biomarkers across large populations. From a given biological sample, these technologies enable us to measure more than 11,000 small molecule biomarkers from a blood sample, from a cellular sample, a tissue, a tumor, or even cells in a dish. Here is an image of our actual RLC suite of instruments that's in the laboratory just behind me. We have a suite of a number of these instruments that really allow us to work very quickly. For each of these molecules that are assayed in a biological sample, we perform what's called 4D chemical classification, including their retention time on our commercial custom columns, their MS1, MS2 characteristics, as well as their CCS values on their ion mobility mass spectrometry. From this amazing amount of data, we can typically identify somewhere in the order of 500 to 1,000 known molecules, as well as capture and measure the tens of thousands of yet unidentified, unknown biomarkers that are present in a biological sample. Now, here at Sapient, we have ways of going back and identifying these unknown molecules using both computational as well as experimental tools that allow us to greatly enhance and grow the number of biomarkers that can be measured and used for human disease understanding. We do this quite quickly. Our laboratories, everything from sample excisioning and handling all the way through analysis on our mass spectrometry systems is fully automated, which allows us to analyze biological samples at great rate and speed. Our typical analytical cycle time is less than 1 minute per biological sample, and this allows us the capacity to handle tens of thousands of biological samples at a given time. Now, these technologies and these robust data generating systems are coupled to an extensive human biological database, which is one of the largest in the world and proprietary here to Sapient. This database encompasses tens to now hundreds of thousands of human biological samples, which have been analyzed already using our mass spectrometry systems. Across the individuals that are included within these biological database, we have decades of clinical adjudicated follow-up that allows for longitudinal understanding of biomarkers in relation to the development of disease over time. In addition, we have orthogonal data, including genomics, microbiome, extensive dietary and lifestyle measures, as well as other omic-based technologies in which we've performed in these biological samples. Now, these two systems, our data generating systems and our database systems, are united with one another through our biocomputational integration. This consists of a data science team with deep expertise in the handling of mass spectrometry data, in the integration of these data with other orthogonal measures, including genomics, in relation to clinical outcomes. And the goal of this approach is not only to generate data, but to rather be able to generate actionable insight that drives forward a drug development program. Now, one such example that we use quite commonly here at Sapient is integration of underlying genomic markers as well as small molecule biomarkers. Within our underlying database that I just mentioned, we have tens of thousands of individuals in which we have deep genomic measures, as well as corresponding small molecule biomarkers that have been captured from blood samples. And we can integrate these two data resources. Shown here on the bottom is a modified Manhattan plot across the underlying genome, looking at the association of common genetic variants with circulating small molecules. What we find is that a huge portion of the underlying genome, particularly on a gene level, actually associates with circulating small molecules. Now, these small molecule biomarkers may bridge the gap between large-scale genetics data that has been generated over the last decade and biological understanding, which has been the great limitation in leveraging genetics data for human drug development. Now, fundamentally, all of these approaches, our underlying bioanalytical tools, our mass spectrometry systems, our database, allow us to do one thing, and that's to begin to align a specific individual with their disease process and a particular therapy that's guided towards that specific individual. When we think about aligning the right patient with the right disease, with the right pharmacology, markers that can be discovered using these systems allow us to begin to stratify patients as part of clinical studies, to understand safety profiling and identify those individuals who are at high likelihood for an adverse drug response. It allows us to identify companion diagnostics in individuals who are going to respond or not respond to a particular therapy, and ultimately to do clinical trial enrichment to allow for much more effective clinical strategies. When we think about the right disease, we can understand biomarkers that reveal disease mechanisms, that elucidate underlying targets that can be intervened upon. We also have biomarkers that allow us to begin to diagnose disease many years or even decades prior to official clinical diagnosis, as well as understand disease progression and understand those individuals who are going to have a very quick progression versus those individuals that are going to have a slower, more occult progression. When we think about therapeutics and pharmacology, these biomarkers allow us to understand dosing and drug-drug interactions, timing of specific treatments, engagement of specific targets, and ultimately pharmacodynamics. I'll show you some examples of these in just a quick moment. Ultimately, what these tools allow us to do, and what this data allows us to do, is to understand clinical heterogeneity, which is the real crux of personalization, both of diagnostics as well as therapeutics. When we think about acquired diseases that are diagnosed fundamentally through pathology, or genetic diseases that are diagnosed through sequencing, we know for both of these that there's tremendous heterogeneity. And what these small molecule biomarkers allow us to do is to subset those individuals into specific etiologies or drug response categories that allow us to begin to personalize therapy based upon specific disease characteristics. Now, I'm hoping we can just take a couple minutes and share with you some examples by which we can do this in the real world. One way we go about understanding drug response is through target engagement, and this is the first example I'm hoping to share with you today. Now, one of the areas that we think a lot about is oncologic therapy. As you can imagine, the PD-1 pathway, which was discovered many, many years ago, has transformed our therapies within the cancer space through a reawakening of the immune system to target cancer cells across a number of solid and liquid tumors. And we've become quite interested, as you can imagine, in beginning to understand this PD-1 pathway. Now, we know clinically there's great heterogeneity in the underlying response to PD-1-based therapeutics in cancer, irrespective of the underlying tumor. Now, there's some data to suggest that tumor burden, mutational burden, or expression of PD-1 may be proxies for response, but we know clinically, once again, that these are relatively poor predictors of who's going to specifically respond to a PD-1-based therapy. Now, using our human biological database, we've probed this underlying pathway. And if we look across the tens to hundreds of thousands of small molecule biomarkers that we've captured in our underlying database, we can begin to ask, are there specific biomarkers in circulation that read out the PD-1 pathway? When we perform this analysis, which we show here at the bottom, we find that there's a handful of circulating markers that statistically robustly associate, I should say, with genetic variants that are present in the PD-1 pathway. Now, if we take just one of these markers that is found to associate robustly with PD-1 genetic variants, we can begin to ask, how does this singular marker perform in response to PD-1 therapeutics? What we find is that this biomarker is increased after treatment with PD-1 therapies, as shown on the left here. So if we look at individuals before or versus after they received a PD-1-based therapeutic, this marker increases in underlying circulation, indicative of engagement of this underlying pathway. Now, even in the post-treatment samples here, you can see there's quite a bit of spread or heterogeneity in which there's some individuals in which the marker goes up higher than in other individuals. Now, if we look at that heterogeneity and begin to ask, how does this map onto treatment response? What we find is extremely interesting. Among those individuals that have the greatest quartile increase in these biomarkers that read out again this pathway, we find there's over a 50% increase in treatment response relative to those individuals that have a more modest increase in these PD biomarkers. This is really a startling discovery that allows us now to begin to map how these pharmacodynamic markers may be influencing underlying treatment response in these individuals. Similar, we can think of other pharmacodynamic biomarkers and ask how they influence our ability to predict drug response. Shown here is a clinical trial that we had worked on together with one of our clients, which was a failed phase 3 trial for a rare disease indication that consisted of several hundred individuals that failed to meet their primary endpoint. Now, if we look at these 250 or so individuals, we can take biological samples from this clinical trial, analyze them using our rapid chromatography mass spectrometry systems, mapping tens of thousands of, again, circulating small molecule biomarkers that are each represented by a little black dot here on the left volcano plot. Now, if we begin to ask, are there any specific markers that change after therapy relative to before therapy? There's one marker here that's circled in blue and indicated as the red dot here that, again, is robustly changing after therapeutic intervention. Now, if we look at the spread in this single pharmacodynamic marker in individuals, what we find is quite interesting and is shown on the top right here, in that there's a population of individuals in which this marker increases after therapy, and there's a whole population of individuals that have no change in this pharmacodynamic marker after therapy. Now, if we take this clinical trial, once again, that failed for meeting their primary endpoint, and ask once again how this pharmacodynamic marker maps onto drug response, what we find is quite startling, as shown here on the bottom right. We find those individuals that have a high elevation in this marker after receiving therapy actually have an incredible survival and appear to benefit from the underlying therapeutic. Whereas those individuals that fail to mount a pharmacodynamic response to the underlying therapy, perhaps indicative of poor target engagement, as shown as the red line on this Kaplan-Meier plot, continue to experience worsening mortality. In this way, we can begin to use these small molecule biomarkers to theoretically rescue failed clinical trials. I'll go ahead and stop there and open this up for question. Here at Sapient, we view ourselves as a discovery organization that works together with our clients to enable their research programs. Our contact information is located here on this slide, and our email is discover@sapient.bio. And I would absolutely welcome anyone who may be interested in these approaches or these technologies to reach out to us so we can have a discussion about how we may be able to best enable your work and your discovery. I'll go ahead and stop there, Stacy, and would be happy to take any questions.
Perfect. Thank you so much for that great presentation. We have a couple of questions that have come in. The first question is, do you believe small molecule biomarkers are more predictive of drug response relative to traditional genomic or protein biomarkers?
Yeah, it's a great question, Stacy, and absolutely there's a role in human both drug development as well as diagnostics for protein and genetic markers. And in no way do I want to discount how transformative these markers have been across a number of different diseases. That being said, we know clinically that these markers have only been able to inform in certain diseases, particularly in the cancer space. They've provided quite a bit of information and understanding particular tumors that may respond to a therapy. But we know that there's many other diseases for which genetic markers or protein biomarkers have failed to really reveal those individuals who are going to respond to a therapy, or to identify those individuals who are going to develop a disease over time. Particularly in these cases, I think small molecule biomarkers can be transformative and may provide an alternative approach for diagnosing these individuals or these specific diseases, as well as identifying those individuals who may be most likely to respond to a therapeutic. Now, the way I very much view this, Stacy, is a complement, that these are not singular systems that are mutually exclusive, but rather they provide different type of information. And by leveraging them together, one can really begin to sort of inform disease diagnostics as well as, once again, therapeutics.
Great, thank you. A key challenge in the small molecule biomarker space is the great diversity. How do you think about handling known versus unknown molecules?
Yeah, you're absolutely right. That traditionally, this has really been one of the great challenges, that while we understand quite a bit about our genome or our proteome, those circulating small molecules in our blood, only a very small portion of have been mapped to date, which means that tens of thousands of things floating around in our blood have never been measured or never been structurally elucidated. Now, the approach we've taken here at Sapient is rather than focusing only on those known molecules, is to say, well, there's many, many things in blood. And as going back to my first slide, we're not really quite certain which one of these is key. And so let's measure them all. Let's let the data tell us which one of these markers is most critical, whether it be a known molecule or an unknown molecule. Now, importantly, for both the known as well as unknown molecules, we've captured these in our human biological database, and we can cross-integrate data that's generated in a de novo sample relative to this underlying database. So there's quite a bit we can learn about even an unknown molecule prior to solving the underlying structure. Now, for those unknown molecules that prove to be sort of important or provide information, and the examples that I showed you just a moment ago, both in the clinical trials as well as for the PD-1 pathway, all of those represent unknown molecules. We have the ability here at Sapient to go back and solve those underlying structures and make them known molecules, both through experimental as well as through computational approaches.
Great. Can you talk through your statistical approaches and how you handle large amounts of biomarker data to identify key signals?
Yeah, so this has been one of the great challenges over the last decade, that as a community, we've gotten much better at generating data. And in many ways, data generation has increased exponentially. Biological knowledge or our understanding has continued to only increase linearly. And the real fundamental issue here is how one begins to take data and make knowledge out of that. As I mentioned, our goal is not to generate data for the sake of data generation, but rather to generate actionable insight that moves forward a program. Now, the way we do that is by applying statistical approaches, as you mentioned. It's critical that we have great expertise in handling very complex multidimensional data, whether it be mass spectrometry data, genomics data, as well as clinical data, being able to integrate this information in a way that allows us to statistically pull out those singular key biomarkers that inform us on a particular disease process or drug response. Now, there's a variety of different statistical tools we use, everything from very complex machine learning and AI-based approaches to very simple reductionist Bayesian statistical approaches, including regression. And so there's a number of tools we can employ based upon the underlying question. Each of these has their own, as you can imagine, strengths and limitations. And as I mentioned, the real goal is to be able to apply these computational approaches in order to bridge that gap between data generation and large amounts of data and underlying biological understanding.
Great. Traditionally, small molecule biomarkers haven't been used to the same degree as genomic or protein biomarkers. Why is that?
Yeah, it's interesting to think about, Stacy. So when we think about laboratory tests, obviously emerging biomarkers over the last several years have really been focused on the genomics as well as proteomics space. That being said, every time you go to the doctor's office and they take a tube of blood, they're measuring a number of small molecule metabolite biomarkers. This includes triglycerides. This includes creatinine as a measure of renal function. This includes glucose for understanding metabolic homeostasis and for diagnosing diabetes, as well as understanding response to particular anti-diabetic medications. And so it's not the case that small molecules haven't traditionally been used. It's just that they have not sort of advanced at the same scale that genomic and other proteomic biomarkers have been sort of used over the last decade. Now, a real reason for that, which I mentioned earlier in the talk, relates to the fact that there has really not been technologies that allow us to measure the thousands of biomarkers present in human blood across tens of thousands of individuals at a given time. And being able to do this across large populations in a very robust, comprehensive manner is really the key, as it was for genomics, in discovering new small molecule biomarkers that then can transform clinical care. And it's my hope that as these technologies are distributed and as these approaches are distributed, what we'll see over the next several years is a resurgence, if you will, in these small molecule biomarkers. Now, I think for certain diseases, going back to an earlier question, they can certainly inform, these small molecule biomarkers will be able to inform more than traditional genomics or proteomic biomarkers, but that remains to be seen on a case-by-case basis. And I expect, once again, that we'll have more of these small molecule biomarkers in the clinic in the very near future.
Thank you. It looks like we have one more. To the rest of the audience, we do have time to take some additional questions, so do put those into the Q&A section on your platform. But in the meantime, how do you incorporate small molecule biomarkers practically into a drug development pipeline?
Yeah, it's a good question. Well, as mentioned here at Sapient, we're a discovery organization that works with dozens of external investigators across biopharma to help advance their drug programs. Now, there's a number of different ways that we can engage and incorporate these approaches as part of the drug development pipeline. We could start very early on in the discovery phase to identify new targets, to validate preclinical models, and to understand disease biology to elucidate new potential targets, as mentioned, for disease intervention. All the way up through these preclinical stage where we're doing a pharmacodynamic marker discovery into the phase 1, phase 2 early clinical studies when we're beginning to understand potential intermediary biomarkers that may reveal response or companion diagnostics. Now, what's really interesting about these tools is they can be applied early on as well as very later on in the disease process. And so there's a number of programs that we work on in the phase 3, phase 4 area, again, to understand response to therapeutics and reveal new targets for the 2.0 versions of drugs. And so it's really an approach that can be applied across the entire spectrum of the drug discovery pipeline, from very early on to more mature phases, all the way up through the end.
Great. Well, Dr. Jain, that is our last question. Was there anything that you wanted to close us out with?
I appreciate your time today. We're excited to work with others to sort of enable discovery. And please don't hesitate to reach out to us if there's anything we can do.
And to our audience, if you want to revisit this presentation or share it with your friends, it has been recorded and will be available on the AAPS website. Thank you so much, Dr. Jain. Thank you, Sapient. We really appreciate, as always, your partnering with us on these great presentations, and we look forward to seeing you soon. Thank you so much. You can all now disconnect from the webinar.